So I did prepare a few questions, and I got some help from from the speakers and the participants of this roundtable. So let me just crack one question at you guys and see where it's going to lead us. So one question is, when we look at your presentation and of course we think about this, I think one question that pops in our head is, are there actually microbiome signatures of healthy versus unhealthy aging? It's a simple question, but I'm not sure we get the simple answer to that. So who would like to start addressing this question? Sean?
If I can, yeah. So I thought about this a lot in the early days because we know the work from Cork, was focused on a lot of these assisted living facilities for a while, right? So you saw that people in long-term care facilities, it looks like the managing the decline side of aging. It looks unhealthy. Things aren't going so well for them. They actually have higher Bacteroides dominance, for example. But then in that same Claassen paper, right, they show if you're community dwelling, then that signature sort of is displaced. And so you can kind of hold on to health better when you're not eating a poor diet, perhaps, or you're around a lot of old people who have C. diff infections and are getting you sick and so on. So I think there are different signatures. What I'm currently struggling with is whether this is a survival effect and how much of it can be changed. And I suspect it can be changed because people going into these long-term care facilities seem to be pushed towards this bad phenotype, although maybe they're just being enriched because they're already sick and they're getting pulled into that system. We have a little bit of data on individual longitudinal stuff where if you look at a person over time, we do see that with healthy lifestyle intervention, you see a little bit of an increase in uniqueness, but we only have like a 1-year time series, and it's not quite enough to have a really strong p-value. So I'm hopeful that you can push yourself in a good direction, but I'm not sure.
Maybe I can continue that.
Please.
I think definitely also believe there's a microbiome synchronous in healthy, unhealthy, healthy aging. But for instance, based on a large scale human cohort study, and we see microbiome synchronous shared across different chronic disorders. So no matter which diseases you have, but actually common synchronous. And these synchronous were not explained by disease comorbidity. So therefore, I really think could be driven by aging-related biological process. However, of course, it's descriptive, right? I could consider the biomarker as you described, that the uniqueness or higher richness, and also shared disease composition between healthy and unhealthy conditions. And doesn't really have to say it's really causality yet, not very hard to establish in human cohorts. Second thing, I think synchronous will be not only see on the compositional level, and also, as I mentioned yesterday, on the genetics level, that we see massive association associated with age. And also very striking observation we found, even opposite way, seems aged microbes seems to have a higher enhanced capacity and functionality to promote good health, because they adapted to the declined physical condition, try to be even more effective. So a little bit opposite to our classical thinking about the microbiome role in aging.
And I could probably add too, again, I'm not involved in this research, but my Cork folks, they also work on signatures and have put lots of effort into it, and they are very focused on keystone species, actually, as in an ecological approach where they say there are, Paul O'Toole would say there are 7 species that are kind of at the center of knots that are then connected, and they are having paper to link. Then what is cause and effect there, again, is clearly not established. Yeah.
Eran?
I agree with all the previous comments. I think 2 important points from my perspective. One, as was mentioned before, in any case in which a correlation or an association was made between microbiome composition and a health or disease status, I think it's clear that the majority are passenger effects, secondary effects. So in the aging process, you have ambulation, exposure to drugs, comorbidities, blah, 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 that probably account for many of these changes, but we're all interested in finding the minority of changes that actually drive disease or aging-related processes, and that's easier said than done, but we'd be very excited to explore. The second point is that we're also finding that some of the variability between studies and within individuals could be deconvoluted by looking more carefully on function rather than composition. So imagine a healthy centenarian in Japan or in the US. You would likely find a different healthy configuration in both populations, but if you start looking at function, it generates a better common denominator to health and disease features. And so I think the more this field will move away from naming bugs and compositions, but by incorporating functions, we would find which functions are present or missed in a given health context, and that would generate maybe more actionable results.
So to jump on this a little bit, to understand function better, because I think we all agree we have to move beyond just looking at numbers of the type of bacteria, but what they do, how they function. And most of you have talked today about using different omics tools, like looking at the proteome, for example. Some would look at the metabolome. So is there something you think, should we look at the multi-omics perspective, or should we focus on one type of signature, which is more functional than composition?
I definitely think multiple omics are very necessary, as we heard a beautiful story from the metaproteomics data, and also metabolomics, as also Sean have mentioned already. I think this is pretty key because, as I said, signals were not easily with only one level of information, only combined it, right? So we had genomic levels that we already probably extensively studied in the past decades. Now metapromics and probably also metabolomics starting rising because a lot of omics data we did on the host side. At a limited actually efforts still behind also in the microbiome side, and how to integrate the host human side and the microbiome side together, I think is another level thing. That Sean mentioned that the genome scale or metagenome scale metabolic modeling, and here maybe we should consider at the whole genome level of metabolic modeling or proteomic network, especially to has a close link between human and also microorganisms. Maybe even consider other non-bacteria components as well, fungi, virus, all together. Right, so this is more comprehensive. I think we get more closer to the true picture.
Yeah, good point. I think we focus a lot on bacteria for several reasons, but what about other organisms? What role they play into, aging trajectories? Should we look more at the virome? Should we look? So, Ruth, you want to?
Well, is this working? Yeah, I think in that sense, I think we all agree that we need longitudinal studies on people, especially if we're looking at aging with respect to younger people. You're looking at people with vastly different histories, colonization histories, might have had H. pylori in the past, this kind of thing, and other organisms too. I mean, from eukaryotes to just different exposures in the past, and that's really difficult to account for. And I think until we have good longitudinal cohorts on aging, it's going to be quite challenging to tease some of these things apart.
Sean, you wanted to add something?
Well, just kind of the multi-omic and like what else are we not measuring context? I mean, anytime you have an interface between two or three things, you're going to find new stuff. I mean, most people have thrown the kitchen sink of omics at most phenotypes, and they've done the low-hanging fruit, which is the individual omic analysis. But finding clever ways to thread together two omics you're always going to find cool stuff there. So I think people should focus on that. And then one of the things that we aren't measuring enough of, brought up by Eran, is diet. So, whether you have a microbiome composition that can do one thing or another is largely determined by whether you ate an avocado that day or an artichoke. And so the combinatorics of having microbiota knowledge and that dietary exposure knowledge and the phenotype data, I think that's going to open up a lot of interesting avenues.
After nutrition or food, and a lot of you have talked about that today, it's always fascinating to see our drugs. When we talk about the exposomes, a lot of the drugs we take are having an effect, a major effect on the microbiome. And of course, if we talk about aging and older populations, there's more and more drugs, right? That probably becomes even a bigger determinant of the microbiome. So how much do you think we, it's like two questions in one. So how much do you think this should be covered better, or should we look at this more carefully? And what tools should be developed? I mean, we talk about personalized nutrition. Should we talk about also about personalized medicine for microbiome and its impact on aging?
Yeah, I think it's an exceedingly important topic, and it's bilateral. It's not just the impact of drugs on the microbiome, which could have downstream health ramifications. Equally important and interesting is the effect of the microbiome on drug metabolism, and I think there were quite amazing studies suggesting that 25% or more of all drugs are metabolized to one way or another by the microbiome. And I think from a bird's eye view, as a clinician in my previous life, I think that if you look on any drug that is given to humans, the most effective drugs always impact a minority of humans. So yeah, the best of the best antihypertensives, would impact 20 or 30%, and we kind of use this knowledge in order to mix and match medications and hope that this trial and error would hit the right person. But I think that revisiting this observation and trying to explain why 70 or 50% of a given population do not respond to a given drug could eventually involve the microbiome. So if we do the same kind of personalized nutrition thought process and apply to drugs, I think we could maybe better harness drugs to the individual.
Sorry, may I add something to that? And definitely I agree what Eran already said, but a lot of area has been overlooked, actually diet-drug interactions. Because while we take drugs, we also get other dietary advice when you should take drugs that actually, and so this is important for the microbiome or in the future health aging is really for the prevention, right? First, the prevention to prevent the development of disease. Also, the secondary prevention to prevent further second comorbidities. So then how to also take diet at the beginning before you have a disease to help personalized nutrition, and then later to combine personalized nutrition with personalized medicine to also prevent the secondary prevention strategies and also improve the drug efficacy and the drug toxicity. I think this is also area we should also take diet, medicine interaction into consideration.
Sean?
Maybe to add a little quantitative flair here, because I have it on the top of my mind from the statin paper. The only reason we looked at statins is because we had this large cohort, and if you just happen to sample a bunch of people in the US, 20% of them are taking statins. So it was just a very common drug, and we had the power to look at this question. I suspect if you looked at any other drug, you're going to see these effect modifications. But for statins, we could explain something like 22% of the variance in the on-target effects in the whole data set. 10% of that variance was explained by age, sex, and body mass index. You could explain about 4 or 5% of the variance with the 9 genomic markers that have been identified from GWAS to be most associated with statin responsiveness. So the genome could explain, 4% of the variance, and then the microbes were explaining something like, 6, 7, 8% of the variance. So, it's on par with the genome. So pharmacomicrobiomics, should be on par with pharmacogenomics.
So what do we do for these people that would benefit from statins because they have, lipid profile, which is pretty bad? And so do we remove them from statin, or are we trying to use a biotic approach in order to change their microbiome so that you don't get the side effects of the drug? Is that possible? Is it something that we may think it's possible very soon?
Yeah, there's actually follow-up work to our paper from China. There's a group, it's Shi et al., I think it's Cell Metabolism, where they put new statin users on the drug, and they looked at things associated with the response. they found that statins were reducing the abundance of certain Clostridia that were metabolizing bile acids, and there was a decline in ursodeoxycholic acid. There just happens to be a drug called ursodiol, which is ursodeoxycholic acid. So they gave a small cohort of statin users, I think it was only like 5 to 10 people, they paired the statin with ursodeoxycholic acid, and the ursodiol completely rescued the metabolic phenotype. So their insulin sensitivity improved, and they still had the same LDL lowering effect. So I think, yes, you can.
But if they'd been on Jens's diet the whole time, they wouldn't have needed any of this, or?
Yeah. Maybe ursodeoxycholic acid is mediating that effect.
That's why the book is freely available.
So we're moving to interventions now. So I think we saw a good example with Jens talk about the Restore diet as a way to really change the microbiome for the better, and for not only to improve health but improve I would say health span, as you mentioned. So now, those that wants to do trials, those that are clinicians in the room, for example, would like to design some trials to test nutritional or biotic interventions in aging populations. So 2 questions. Should they do a study with people that are 40, 50, 60, 70? When should we start doing these interventions? And what should be the design of those studies in order to make sure we get the most out of these research, these trials? May I start with you, Jens?
As I showed this observational study from the UK cohort, they looked into this, and their model said, if you start eating healthy in your 40s, it's 10 years you can add, and if you start in your 70s, it's 4 to 5 years. So I guess it's never too late. But I guess the earlier you start, the better it is. And then it really just depends on what you try to achieve with your trial, yeah. In terms of how to design it, even that's a world in itself, that's the nutrition. We in the group, we are very, Eran mentioned how hard it is, to make people eat a certain way or to even just measure it. So we just try to get away from this as much as possible, and just we try to control. We don't do the Kevin Hall, we don't put them in prison, for 4 weeks, but we do hand them every single meal, which is a really good experimental design, because so there's actually no incentive really to cheat, because they would have to spend extra money. They get the food for free, they save time, and from what we see, we have very little indication that compliance is not very hard. But then there are any other experimental design, you can advise them, you can just look into a cohort, you can try to measure it, so it's hundreds of different ways of doing it, yeah. But we personally, in our group, we try to control as much as possible. But that comes then with the disadvantage that they are shorter, because you can't do this for longer than 2 months, for example. So if you would want to go a year or two, then you would need a different experimental design, yeah.
So if we go from diets now to biotics, so what should be the approach? I'm going to take the side of the prebiotics, just for the sake of discussion and debate. So with prebiotics, at least you can design the prebiotic to act on a niche or a very specific organism that will be beneficial. There will be probably 10 or 20 microbes that will benefit from it. So the chance of success is maybe greater than for a probiotic, for example, or even a cocktail of probiotics. Do you agree with me, or you think both of them have their chance to have an impact? Who wants to take this one?
Well, I respectfully disagree.
Okay.
I agree and disagree. I think you probably did this for the sake of question, but the reality is much more complex than just comparing one to the other. And I'm not going to start dwelling on it, but even if you look at the most simple prebiotic fiber-based intervention, I would argue, and I'm arguing based on data that we've generated, that not all fibers are created equal. There is huge divergence in the chemistry of different fibers. So expecting that a prebiotic would do something very uniform, given this divergent chemistry and the different effects on so many species, I think is maybe romantic, but less evidence-based. And we need to do much more work in order to refine what we know about these effects. With probiotics, don't start me. Let the others talk.
Okay.
Yeah.
So I can add to this. So we actually, when in the group, when we did the non-industrialized type diet, we had a parallel study on fiber supplements, and it's pretty in agreement. So while we had very, very low inter-individual variation with the complex diet, probably because they eat all kind of different foods, we had very, very large inter-individual variation with the single fiber. So there's a lot more than rational to individualize this. And I would also, let's say the clinical effects were rather minor and perhaps also disappointing. So I think when it comes to dietary fibers, I have more hope in symbiotic probably approaches, and then probably also, more complex one with mixtures of fiber, perhaps mixtures of microbes, and there I'm a bit more hopeful, and then also go for specific mechanistic targets to try and achieve something.
Yeah, sure.
So we've done some modeling of some of these prior intervention studies to try to understand, responders, non-responders, from the microbiome metabolism context, and we definitely see a lot of variation there. It does appear that, you give 10 people a banana, very different chemicals come out the other end. So even for the exact same intervention, you're getting very different effects because of the microbiota. But I think we can target things. So if you could get to the point where you could pre-partition people into responders and non-responders to a given fiber, then you could just deploy different fibers to different people. To the mix question, we also find that if we mix fibers together in the models, we can reduce the average non-responder rate in the population. So mixtures, at least in the simulation context, appear to be more robust, and I think that fits too with like a complex diet being more robust. You're hedging your bets across a lot of different things. So whether you're doing something really difficult and complex for someone to adhere to possibly, or you're trying to do something more simple with a fiber mix that might, or precision targeting.
Yeah, I think we talked about personalized nutrition, personalized medicine. Here, I think we should not forget personalized microbiome therapy. Because as many clinical trials already show, the baseline microbiota factors actually determine the microbiome targeted or dietary targeted or probiotics-based approaches. And so how these probiotics or with the prebiotics, how we help these species can be colonization in your gut is a big question as well, because most of the prebiotic trials doesn't really have a long-lasting effect. So because it cannot colonize, so then we have to need more evidence to have a more ecological point of knowledge and a basis to help what is the most efficient and long-lasting effect we can achieve.
Okay. So diets, personalized diets. So if you would have to name the 3 foods that are the most important to drive changes that are beneficial and can help to improve healthy aging, what would there be?
Red wine.
Well, that's a fermented food. Something that hasn't come up in this discussion yet is fermented food, which is one of the original ties to longevity with yogurt. I don't know how much people have looked into fermented foods in the context of longevity, but that seems to be something that also increases diversity, possibly through the metabolites that are in there, also possibly delivers probiotics as well.
Yeah, fermentation in general, for dairy product in particular, has been shown to reduce the risk of type 2 diabetes and other related disease. And of course, all these chronic diseases are linked to aging. So, but I agree with you. I don't think I've seen really good studies that have looked in that context. Really.
So I think there's a lot of excitement around it, and in the nutrition field and in Cork, we have several groups looking in it. But when you then look at the actual, human trials data, there's still very, very little. But I think this is hopefully going to happen now. Yeah.
But now fermentation, fermenting carbohydrate is good. Is it the same for proteins? because I've seen a lot of presentation today at this symposium and others talking about some fermentation of some proteins will lead at least to the formation of maybe metabolites we don't really want. And it's very debatable. I think, Sean, you talk about phosphocrystal sulfate. Is it good or bad? It's same for TMAO, actually. There's a lot of debate. Yeah. So is fermentation good for all type of food or not?
Though I do not really know fermented meat products good or not, because I don't think we had much scientific evidence yet, but I'm really happy you raised the questions regarding the protein, because I think we have a lot of studies talking about the fiber, it seems, but I think they already can see that actually diet, protein and studies in terms of the human health has been under-evaluated, because a lot of products metabolize relate actually amino acids metabolism, like tryptophan and thermal, that is a carnitine, and also arginine. They all link to all kinds of products, actually linked to all kinds of diseases. So all related to the protein intake, branched amino acids, a lot of amino acids metabolism. So I really think this is a big area. We should learn even more. In addition, short-chain fatty acids.
And there's quite some controversy in this, actually. When we wrote our review on healthy eating and the microbiome, one of the authors is Carla Brado. She's from Brazil. It's very hard to convince a Brazilian, that meat is detrimental for you. Yeah, so there was a lot of, that was actually the one point where we had the most discussions. And obviously there's a lot of benefits. Actually, you mentioned it, I think, then and they have to, it's very context. It's very hard to make some strict, points, because then if you get into a frail, and that's Carla works on sarcopenia, you don't want to recommend low protein diets, to these individuals. But then in a longer term and throughout life, I think high protein diets are, at least that's what the nutritionist thinks, are more detrimental. And in mouse studies also, they do shorten life. But it's very, very important to have nuance there. Yeah.
Yeah, it's also like now the foodomics become more a rising area, right? Because also we are human beings. If you said we only eat one type of food, probably most of us say, no, I cannot live with that. I would rather enjoy from my life. So like a food diversity is another component, right? You should actually, like we had the microbiome diversity, maybe you should take also a larger variety of the food to enrich your diversity and enrich your metabolism and all kinds of the compounds. So not separate. Yeah.
And I still want to do a Korean barbecue tonight.
Yes.
Yeah. With kimchi then.
Yes. We also want enjoying our life as well. The happiness from food is also important for healthy aging.
And that's,
I think from the, oh, sorry.
No, as I say, that's one of the clearest results you showed, Jens, is that red meat is really bad. And there's all these people trying to push the carnivore diet out there, these influencers. Yeah. And I think that's just terrible for you. But it is complicated. Like we see, indole is kind of maybe good for you. It's anti-inflammatory and it's derived from tryptophan. But then there's a lot of others that seem to be bad. If you're constipated, you seem to have more of these. So it looks like there's a switch in the gut. Like normally your microbes will produce short-chain fatty acids from carbohydrates, but if it's a hot potato, right? If it sits around for too long, it runs out of substrate and starts to ferment proteins, and then you get this buildup. And that's been associated with chronic diseases like kidney disease or dementias. And then I've seen some evidence from trials where in young people, having high protein maybe is bad for you, but it really depends on if it's plant-based or animal-based. But if you do then that same intervention in like an older, frail population, suddenly you see benefits because you're protecting people from sarcopenia.
And I think the discussion about proteins is always low versus high protein diets. I think we don't think enough about looking at the composition or the different type of proteins in our diet for just a regular amount of protein in our diet. I think it's as important as to look at different type of sugar or of course lipid in the diet. I see there's only 1 minute left, so one question I have quickly for you guys. Most of the talk today were about the gut microbiome, but is there surrogate regions that we can use? What about the oral microbiome? Some of you touch about it. Some would talk about the skin microbiome. So sometimes these low-hanging fruit type of sites can be also very important. So what are your take on this in maybe 10 seconds for each of you? That's okay, 15 seconds then.
Yeah, so I'll take my 10 seconds. The answer is these extraintestinal microbiomes are exceedingly interesting. These are niches in which the microbiome in each niche has co-evolved to generate functions that are unique to the niche. They're more complicated to study. They're usually lower biomass. The skin is one example. But also present opportunities to explore niche-related diseases that are neglected if we just look at the gut. So we and others are doing this extensively, but it's easier said than done in some cases.
And my second is that all area of course important regarding aging. I think microbiome translocation across body side will be very important because skin microbiome can easily enter your mouth and the mouth can be translocated to your gut. And so this transmission, how they migrated, how they at a different niche, what function they're going to play, whether they cause disease or not, I think it's a dynamic map for microbiome that will be very interesting to investigate.
So the oral microbiome, when it relates to diet, is already important because of dental disease, which obviously is driven by sugar content in the diet, and then that has downstream effects. So it is obviously, we are not looking at it, but it is an important.
But it's relevant, yeah.
Yeah, I agree. I mean, it's the oral microbiome has changed a lot with the Neolithic revolution, and it's created a lot of problems for our ancestors, and probably continues to do so for us today. It's definitely a health and diet readout.
Thank you.
I'll probably keep focusing on the gut, but I am excited about immunomodulatory effects in other compartments. And there's tantalizing work from the Fischbach lab, right, in mice that you can affect systemic immunity just by painting a microbe on the skin. So I think that work's really fascinating.
All right, thanks you guys for staying on time. And so that closes the symposium today. I would like to thank again the Danone Institute for sponsoring this event, all the speakers and the participants, of course, to the roundtable. And thanks to you for showing up and asking great questions as well. Thank you.
Thank you.